Hello everyone and welcome to today's live webinar, Transcriptomics to Decipher the Pathobiology of Drug Resistance and Enable Future Targeted Therapeutics, presented by Dr. Yesim Gökmen-Polar, an Assistant Research Professor in the Department of Pathology and Laboratory Medicine at Indiana University School of Medicine. I'm Susie Valdez and I'll be your moderator for today's educational webinar, presented by LabRoots and sponsored by Thermo Fisher Scientific. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click on the green Q&A button located at the lower left of your presentation window. Type your questions into the box that appear on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you're viewing this presentation in a slide window. To enlarge that window, click on the screen icon located at the lower right. If you have any trouble seeing or hearing this presentation, click on the support button at the top right of the presentation window or use that Q&A button and let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Click on the button in the bottom left-hand corner and follow the process to obtain your credits. I'd now like to introduce today's speaker, Dr. Yesim Gökmen-Polar. Dr. Gökmen-Polar is an Assistant Research Professor in the Department of Pathology and Laboratory Medicine. Her research centers on mechanisms of resistance to standards of care therapy and the identification of novel therapeutic targets in breast and thymic cancers. More specifically, her work has emphasized targeting of FA2 receptor tyrosine kinase, protein kinase C, beta, anti-tubulins, and TORC1 and 2 in colon, breast cancer, and thymic malignancies. Her lab developed and characterized the only human Thymoma cell line, IU-TAB1. Dr. Gökmen-Polar's current research uses next-generation sequencing, also known as NGS, next-generation microarrays such as Clariom D, human transcriptome analysis, and preclinical models to understand the transcriptional regulation mechanisms of resistance and develop novel targeted therapies. The ultimate goal of her research is to prevent development of recurrences and improve the quality of life for all patients. Please join me in welcoming Dr. Gökmen-Polar. I will now turn the presentation over to her. Welcome, Doctor.
Thanks, Susie, for this nice introduction. Before I start, I would like to thank Thermo Fisher Scientific and LabRoots for enabling this webinar. As you see from the title, I will talk about the importance of transcriptomics to understand the biology behind the drug resistance in cancer and develop future therapeutic targets and strategies for this disease. Let me give a brief introduction. So the main failure to the therapies may be due to the resistance. So the resistance can be in acquired form, which occurs after a drug treatment after a certain time, or it can be pre-existing at the beginning of therapy, it is already resistant. Of course, there are multi-factors for drug resistance happening. And most of the time, because the cancer is very heterogeneous and we don't have enough knowledge about each cancer subtypes and also different targets, and therefore specific treatments are not available. So what are the tools to understand resistance in a preclinical research studies? Of course, we follow the central dogma of molecular biology, meaning that in a very simplistic form, DNA makes RNA, RNA makes proteins. So the information from DNA is transformed to the protein. And there may be alterations at the DNA level, like mutations, which we detect with the DNA sequencing, or there may be differences in the gene expression at the messenger RNA level. We have RT-PCR and conventional microarrays, and also ultimately the protein is the gold standard, and we measure the protein expression where the enzyme assays, immunohistochemistry, and other assays are coming. So these are all fine, and the last 2 decades especially, we used to use these techniques. However, with the introduction of the next generation sequencing, our view is immensely changed. Mostly, we thought always that coding regions, coding RNAs are the most important, and they are translated to the proteins. However, these new technologies give us the complexity of the RNA world. Namely, there are non-coding RNAs that we don't know what they are there for, or there are multiple splice variants we don't know again, although they don't code some of them a protein, they are still have some function. So with these new technologies, we started understanding that it is much more complex than we until now know. In that sense, we started analyzing the non-coding RNAs and then the splicing factors or other splicing regulatory proteins in breast cancer progression and resistance, and published some papers. This also changed the understanding in molecular pathology of breast cancer and their contribution, especially at the regulatory level. So let me give a brief introduction for the non-coding RNAs. Non-coding RNAs usually classified based on their size or structure. If they are less than 200 base pairs, they called as small non-coding RNAs. If they are greater than 200 base pairs, they are named as long-coding RNAs. So among these non-coding RNAs, especially, you can see here, microRNAs are the most established and well developed in cancer. And the other ones, like siRNAs or pvRNAs, small nuclear RNAs, small nuclear RNAs, they are still in their infancies. On the other hand, long-coding RNAs can be classified based on their location. It can be intergenic, intronic, they can be antisense, or others. Of course, there are, for the small non-coding RNAs, ribosomal RNAs and transfer RNAs that we always thought they are involved on the housekeeping processes and never thought in the context of the cancer. However, alterations of those genes, whether expression level or other aberrant structural alterations, also may involve in the cancer and drug resistance. So with this slide, I just would like to give some examples for long-coding RNAs in breast cancer. They are mostly classified based on their function. So on the upper side, you see they can regulate chromatin modification. Hot air is one of the best examples for this in cancer and metastasis. And MALAT1 is another one involved in the nuclear organization. Of course, there are others like act as a protein or mRNA decoys or SNO RNA host, or they involved in multiple signalings that affect the cancer mechanisms and drug resistance. Also, the next generation analysis, sequencing analysis, evolved our understanding in terms of importance of alternative splicing. Mainly, splicing is an RNA editing, you can see on the left-hand side, where the introns, the intervening sequences, can be separated from the exons, the coding sequences. The premature RNA is converted to the mature RNA, having exons only, and this further translated to the proteins. And of course, splicing can happen in different forms we call alternative splicing events. Here you see the basic 5 basic events, and the different splicing will end with different protein isoforms. At the beginning, on the right-hand side, you see the exon skipping, where the exon can be skipped or it can be included. Another one is mutually exclusive exons. 2 exons can be skipped or included, or one each other. Or this splicing can occur in the 5' donor of the exon, or 3', or an intron can be retained. So all of them can end in different functions. The function at the end could be similar or opposing. Therefore, very important to understand their importance, especially in cancer and compared to drug-resistant forms. So here, I would like to give a very brief summary. Also, it has been established well that alternative splicing or splice variants are very important in every step of cancer hallmarks, initially introduced by Hanahan and Weinberg. As you see here, you can see in the proliferation or growth, evading growth suppressors, angiogenesis, immune system, whatever the hallmarks is, you see these are alternative splicing variants that can make a big difference in the function of the process. So what is the most appropriate technology to study all these transcriptomic changes in preclinical research studies? Well, here on this slide, I just wanted a brief information about how it used to be. Like the last 2 decades, we were so excited about the conventional microarrays. We could do gene expression, we can see differences, and there are multiple differences exist when we do a microarray assay. And however, of course, they are limited in terms of their gene expression, and they can be biased, what kind of, if it's whole genome, or it used to be like 500 genes, you choose it. So it depends on what you do, but it was biased. Of course, with the introduction of the RNA sequences, this whole concept changed, and then we were so excited about that we can detect some novel transcripts, we can detect some non-coding RNAs or alternative splicing variants. However, this also depends on the depth and coverage. So here, recently, the next generation microarrays are introduced. So how did it, if I tell the difference between them, so the very original exon arrays, 3' exon arrays in the bottom of the right-hand side, the figure, you see that most of the probes are focused on the 3' end. And gene arrays, you see the probes in the exons, they were not that covered. But with the introduction of the Clarium assays, here you can see the Clarium S assay, you see extensive probe coverage for the exon. And for the Clarium D array, the ultimate, you can detect the overall transcriptome analysis. And you can see multiple exons, multiple probes for the exon and for the splice junctions. As you see on the left-hand side, this is because of it was developed by combining 15 data sources listed here, and that extended or expanded the coverage of both human coding and non-coding transcriptome. If you look in a more detail to the next-gen microarrays, here on the right-hand side, you see the actual Clarium D array, and then on the left-hand side, it explains why they are so extensive, because for the exon, per exon, you have 10 probes, and for the splice junction, you have 4 probes covered. That provides extensive coverage for the both low abundant, high, or alternative splicing variants. So let me give then a comparison between Clarium D and RNA-seq, since these are the methods that you would like to choose when you want to do the whole transcriptome analysis. So for RNA sequencing, if you look at the depth, if you choose 30 to 50 million reads, you can only focus on the high expressors. So it's very much depth dependent. If you increase this to 100 million reads, then 32% of the low expressors there. And even for the long non-coding or alternative splicing, you have to really increase your depth to even 300 million reads so that you can detect all these transcriptomic changes. On the other hand, for Clarium D, it's just one assay. So because of the extensive coverage of both human coding and non-coding genes, you don't need to worry about the which depth I need to choose. You can detect high expressors with one assay, low expressors to the level of 1.5 to 3 transcripts per cell. You can detect long non-coding RNAs, alternative splicing, and anything or novel transcripts, anything you want, because of you don't worry about the depth. But with RNA sequencing, you're limited because for every purpose you have to use a different depth, which it kind of complicates the situation. So also another advantage I like is the time to results. If we do usually Clarium D like 3 days for experiments, and then you have the tech transcriptome analysis console, and you import your data and you immediately start analyzing your data and going back to the wet lab and coming back, so you can parallelly use both of them all the time. As a molecular biologist, it's a big advantage for me compared to the RNA sequencing. You have a really dedicated bioinformatician to analyze the data intensively and then give to you, which is very time dependent. Also, another advantage of Clarium D is if you compare with RNA sequencing, so no ribosomal RNA depletion is for whole transcriptome gene is necessary, where in the case of RNA sequencing, you have to deplete it. And if you want to analyze different samples, like blood, you have no globulin reduction, or also from time to time we uses FFPE samples, formalin fixed paraffin embedded. They are very challenging samples. There is a Pico kit available that also detects very low amounts and also challenging samples, which is very challenging for RNA-seq. Of course, also Clarium D is very cost effective compared to the RNA sequencing. So with that, I would like to present to you 2 projects from my own preclinical research studies. As Susie said, my focus is thymic cancer and breast cancer. So the first project will involve the thymic cancer, and then trying to identify therapeutic targets in thymic epithelial cancer. Let me give a brief background for the thymic cancer, mainly Thymomas and Thymic Carcinomas. They are rare cancers of thymus gland. Thymus gland is a gland that is important for immune function mostly. So it is 1% of this gland is epithelial cells, and the cancer develops in this 1% in epithelial section. So according to the World Health Organization, WHO, they have classified, the Thymomas classified into 5 different histological subtypes. As you see here, A, AB, B1, B2, and B3. And Thymic carcinoma used to be type C before, is a separate section or separate cancer. But the Thymomas usually are more indolent than the thymic carcinoma, and however, all type of Thymomas can spread to pleura or can metastasize, but their speed is different. On the other hand, type 3 Thymomas and also thymic carcinomas, they are the most aggressive tumors, and they are the most types that are resistant to existing therapeutics. So, of course, because it's a rare cancer, therefore there are lots of limitations in terms of diagnostic and treatment strategies. So usually, as a standard of care, these therapies are from the other cancers, like mostly lung, because it's closeness to the thymus, although they are totally very different in terms of the biology. And therefore limited novel biology therapeutics existing, and we usually using just the standard care of therapies by now. So in order to understand the Thymoma and Thymic Carcinoma, we studied the molecular profiles in our preclinical research, and so we developed a gene signature to determine the metastatic behavior of Thymomas, and also to distinguish between Thymic carcinoma and from Thymoma. So here, the left-hand side, the Venn diagram shows the gene signature, the overlapping genes in Thymoma and Thymic carcinoma, also the unique ones to Thymoma or Thymic carcinoma. So we decided to do a protein-protein analysis to determine which gene or gene signaling would be the driver for this cancer, and then we can develop or find some therapeutics to test. On the right-hand side, you see the analysis of protein-protein interaction. Mainly, we ended up with 3 hubs. As you see, the drivers for each hub, the genes, STC2, JPH1, and SLC9A2, and they are all connected to each other by a protein called UBC. Mainly ubiquitin C pathway is involved, and this UBC is mainly regulates the protein turnover. So then this led us to the drug called Bortezomib. It's a proteasome inhibitor. It's mostly in use for the multiple myeloma based on the disease mainly developed on this biology. And we have 2 different models, cell line models, to test this drug. And IUTAB1, it's a Thymoma cell line, indolent Thymoma cell line that we have developed in the lab. And the second model is Thymic Carcinoma model, which is very aggressive therapy-resistant cell line. So we chose these 2 models to see the differences between both biology and in terms of the therapeutics. On the left-hand side, you see both of the cell lines are resistant to cisplatin. Cisplatin is used as a standard of therapy in this disease. So it's the IC50, the inhibition at the 50% cell inhibition is 11 micromolar or 19 micromolar, which is, that means highly resistant. Then we tested the same cell line models to Bortezomib, and you see that the Thymic carcinoma is highly sensitive to this drug, about 200 nanomolar levels, which is very hard to have a nanomolar level drug response. On the other hand, the Thymoma cell line didn't reach the IC50, which was okay. So we are excited to see a difference between these models by using this drug. So then we decided to do Clariom D analysis to look at the transcriptomic basis for therapeutic response using the TAG4. It's the new version of transcriptomic analysis console, and understand the biology behind. Here on the right-hand side, you see the workflow. Mainly, you import the cell files, which are cell expression intensity files, expression files, and then you can perform either gene level or exome level or alternative splicing level analysis. And then these analyses are very interactive, as I want to show in the upcoming slides. And also another aspect or characteristic I like is you can connect the location or exon location or gene, whatever you want, easily to the other databases such as Ensemble, NCBI, or UCSC. That makes you understand further the biology in a very short time. So we first looked at the gene level data to compare the differences of Thymic Carcinoma with the drug treated or non-treated, or Thymoma with the drug and non-treated conditions. Here we have identified 489 genes for the Thymic Carcinoma cell line, and then 279 genes when you treat the cells in response to Bortezomib. In this slide, you see a different level, like the pie charts will show you the group. For example, for the upregulated genes, we have different groups. It tells you whether it's a coding or non-coding or a multiple complex gene. Here, multiple complex means it's very complex. It has both coding and non-coding part. So it is interestingly, the coding region, coding genes are very, very small, 11%, and most of them are either multiple complex or non-coding. And the same for the down-regulated one. Another graphic view is you can use volcano plots, and then you can click these individual dots. Upregulated means for the red, and then green for the downregulated. When you click it, it tells you, it connects you to the tabular format. Here is the gene list. You can choose whatever filter you want. Of course, the default is the p-values or fault change. You can use the FDR, false discovery rate, or you can use from the drop-down menu customized annotations in the upper corner that makes you whatever, there are more features that you can use and to understand more in detail. And then the groups, of course, showing coding, multiple complex or non-coding, and gives you an brief overview about the gene that is downregulated or upregulated. Here I show an example. You see the highlighted gene HSPA1 or HSPA1A. So these heat shock proteins are upregulated in response to the Bortezomib in the Thymic Carcinoma cell line. So I especially chose the upregulated ones because usually with the downregulated, you see that most of the oncogenes or other antiproliferative or proliferative genes are downregulated. But in this case, heat shock protein is upregulated, so usually they are protected from apoptosis. This also suggests us that this can be a potential drug resistance mechanism for Bortezomib when we use in vivo, and therefore it is highly guiding us that we need to also test the combination of Bortezomib with the inhibitors of targeting the heat shock proteins. So it gives you further idea behind this mechanisms of sensitivity versus insensitivity. Here you see an alternative splicing view, and I continue with the gene view, and here actually I filtered with the non-coding genes so that you can see that you can also filter based on the group, non-coding or multiple coding or anything. So here I chose the hot air M1, which we know from the other cancers. There are other novel transcripts, but we will focus on the hot air M1 as its importance in the drug resistance for other cancers. So then we decided to focus also study alternative splicing events and in Thymic carcinoma in response to Bortezomib. Here you see that the distribution of the alternative splicing events. So the majority of events, 62% about intron retention, and that follows by the cassette exons, about 22%. This is interesting because most of the time in other cancers we have the cassette exons the most important affecting, but this drug is important for alteration in tron retention. So this gives us the level of what events are important. Here is the alternative splicing view, namely the blue boxes on the right-hand side. You can see that the splicing is the exon splicing is based on the two algorithms, and then these algorithms means that how much exon expression level is changed between the two conditions, in this case drug-treated versus non-treated, and after you subtract the gene level changes. And exon splicing index giving you the level of the fault, it's high or low. And of course you can also filter based on the exon p-values, FDR p-values, and you can look at the exon event scores. Mainly it's a scale from 0 to 1, and if it's closer to the 1, it tells you how likely is the splicing event in this case. So here's an example for on the left-hand side, the splice variance visualization. You can see that for each of the conditions, sorry, if it's a little bit small, the letters, but for each cell line, it shows you the individual intensity, blue or yellow, and then you can see the signal levels, sample level signal levels, and also on the bottom you see in the box the very red and then highlighted the splicing event levels, either red or green. So it gives you in detail which exon or PSR, probe selection region important, and you can see here, but in the actual software you can click the box, the red box, and it connects you further to the other databases such as Ensemble or UCSC browser based on the chromosomal location, and you can immediately connect yourselves where you are or other biological significance type of related analysis. On the right hand upper side you see the RNA isoform level visualizations, and then here you can see the ensemble transcript numbers, and if you look at the highlighted blue line, you can see this particular exon or PSR is present in some of the transcripts, but not all. So it tells you which exon number is included or excluded based on the transcript isoform. So after this analysis, we decided to look at as a complete what else is changed besides the proteasome system pathway, because this is the main focus or mechanisms of action of the bortezomib. So we also see that alterations in other pathways such as nuclear receptor meta pathway, VEGF signaling pathway, circadian rhythm related genes, MAP kinase signaling pathways, which is very exciting that we will go all after these, because most of them have also potential therapeutic agents available. As a conclusion of the first project, we identified the ubiquitin proteasome pathway as a key hub in thymic tumors, especially in thymic carcinoma, and bortezomib has a therapeutic utility in thymic carcinoma compared to the Thymoma. And at the transcriptomic level, we have identified transcriptome isoforms that as a next step we would like to understand further their biological significance, and also we would like to as a next step perform in vivo studies, in particular combine bortezomib with the heat shock targeting inhibitors to improve the efficacy and to prevent the potential resistance for this drug. So I'm going to switch gear to the second project that is involving the understanding of the endocrine resistance in breast cancer. So let me give a brief background. Breast cancer is a very heterogeneous disease, and endocrine therapies is most of the therapies, especially for the breast cancer that has estrogen receptor expression available. So namely estrogen breast cancers, about let's say 60–75% is driven by the estrogen receptor. So therefore it's important to detect whether it's there or not, and if there's an estrogen receptor expression, then definitely endocrine therapy is the first choice. However, despite the successes of endocrine therapy, there is still resistance is a big challenge. So it can be in different forms. As I mentioned earlier, it can be intrinsic or acquired. And tamoxifen, the most heavily studied drug, about 50% resistance as a first-line therapy. So let me give a brief summary for the types of endocrine therapy available in breast cancer. So it can be a selective estrogen receptor modulator. Tamoxifen is a good example for that. It's an antagonist for ER. Mainly it competes with the estrogen, the ligand for the estrogen receptor. And then because of the competition, it's antagonist, but it can be some agonist. So it's kind of not complete inhibition, but still a very good inhibition. For the aromatase inhibitors is another class, which inhibits the enzyme aromatase, which synthesizes the estrogen. So it's an indirect inhibition of estrogen, but it's especially very successful in postmenopausal women because of the estrogen is driven based on the aromatase rather than estrogen itself. And then the third group is the estrogen receptor downregulators, mainly those drugs downregulating the estrogen receptor. And Faslodex or fulvestrant is a good example for this type of drugs. And there are, of course, multiple mechanisms of endocrine resistance have been reported and postulated and suggested, especially in preclinical research models. And you can see on the right-hand side, most of them, they can be canonical or non-canonical, meaning that if it's a nuclear genomic pathways, that means that it directly affects the estrogen receptor, which is in the nucleus as a transcription factor, or it can be based on independent of estrogen receptor. That means that there are alternative pathways have been introduced, mainly the growth factor receptors, EGFR, HER2, IGF-1R. These are the mostly established drug-resistant mechanisms. So to further understand the whole transcriptomic profile of endocrine resistance, we decided to use the well-established drug endocrine resistant breast cancer cell lines. Mainly those cell lines are derived from the endocrine resistant, endocrine sensitive parental MCF7 cell line. It's an ER positive breast cancer cell line, and LCC2 is the cell line that is treated with the drug tamoxifen and with time acquired resistance to this drug. And LCC9 is the fulvestrant resistant cell line, which is treated with the fulvestrant and confer resistance within certain time. And it's also found out that tamoxifen cross-resistant too. And besides these two models, we also have preliminary data that a gene called ESRP1, mainly epithelial-specific regulatory protein 1, is also important in endocrine resistant cells. So therefore, we decided to stably knock down this and understand the transcriptomic profiling. So here is the data that we have looked at using a QRT-PCR assay. ESRP1 expression is elevated in endocrine resistant cells. So you see that the light blue is the sensitive MCF7 cell line. If you compare the resistant cell lines with that, and you see that huge dramatic overexpression of LCC2 cell line, which is tamoxifen resistant, dark blue, and LCC9 and fulvestrant resistant, purple. So they are overexpressed compared to the sensitive MCF7 cell line. So let me give a just very brief introduction to ESRP1, which is epithelial splicing regulatory protein. It's, as you see from the name, it's epithelial cell type specific splicing factor. It has been shown that it controls splicing of genes important in epithelial mesenchymal transition, which is called EMT. And this is important for invasiveness of cancer cells, including breast cancer. So it regulates alternative splicing events, mostly alternative cassette exons. And however, it's not that much biological relevance known in breast cancer and their potential clinical outcome. So therefore, we decided to analyze this gene. Here, what we have done, first, we stably knock down this gene in these two endocrine resistant cell lines. The upper panel shows the QRT-PCR verification. As you see, the black is the resistant format with the empty vector, and then you see the two clones, light blue and medium blue. So the 2C3 we chose based on the maximum inhibition of this gene. And for the fulvestrant resistance on the right-hand side, again, the resistant compared to the two clones, the lighter colors, 9C2, the maximum inhibition. So we chose those two clones to further undergo or functional and transcriptomic analysis. Here on the lower panel, you see we introduced them to the mammary fat pad models, which is important for breast cancer analysis. And then with the, you see a huge dramatic difference in the tumor growth on the left-hand side for the clone 2C3 versus the resistant version. And on the right-hand side, the 9C2, which is the fulvestrant ESRP knockdown versus the resistant. So this further led us to understand the alternative splicing differences first, because ESRP1 is a splicing factor, so we expect that it's gonna important to regulate splicing of the genes, and therefore the alternative splicing events to look at it. Here you can see, as it's as I earlier said, that it affects the cassette exons, mainly skipped exons or inclusion of exons. In this table, we also, as significant events, the majority of the events were belonging to skip exon or cassette exons, being 348 different exons corresponding to 289 genes. And you see the other events also existing, although not dramatic as the others, but still existing. So here you see a genomic view of this analysis. This is the gene, we looked at it as CD44. And I will show this gene because it's a well-established gene in terms of an alternative splicing, importance of alternative splicing variants and biological relevance. Here you see on the left-hand side the ensemble transcript numbers, which you can see which transcript, which isoform is having these multiple exon inclusion versus the ones that they don't have it. So here in the very top, the PSR exons or probe-specific regions, you see the red boxes. All of them are excluded in certain isoforms and not excluded in others. But in the resistant form, it is included, and in the ESRP1 knockdown, we don't see it. So there is a difference between resistance and then the non-resistant one. So here in this slide, actually, I would like to show what this means in terms of the biological importance. Exon skipping or including alters the function of the CD44, and this also makes it confer to the resistance. So if you look at the upper side, the exons from 5A or 14 depends on what type of tissue or cell the exon number may change. However, here, if it's included, you have, it's called CD44 variance form. If it's excluded, you call it standard form. And if you look at the right-hand side, you see the structural view and what it means in terms of protein domains. So here the cytoplasmic, on the CD44 standard, you see the cytoplasmic domain, transmembrane domain, and N-terminal domain. On the other side, with the variant form, you see that there is an extra, they call it stem, here in the N-terminal domain. So this extra form, which is coming from the inclusion of the exons, actually changes its function. So it makes or confers resistance and make it more invasive or important later for metastasis. So as a conclusion from this study, we did the transcriptomic analysis and identified significantly altered alternative splicing events in knockdown of ESRP1 gene. And as I give in the CD44 example, alternative splicing alters the function in the cells, and it either makes it resistant to the therapies or it changes the sensitivity versus resistance of the cells. And so the next step, we would like to test the different therapeutic agents to switch the alternative splicing events, or in the case of CD44, can we switch these variant form to the S form, and for the other ones, that's our next goal to perform. So as an overall take-home messages, what I would like to say is, of course, the goal of the study is important. You can have a discovery versus targeted approach. You may focus on high expressors, or you want to look at the low abundance transcripts, or you would like to do alternative splicing. Whatever you choose, I think the importance is nowadays, of course, you would like to look at the whole transcriptomic analysis, because those non-codings or alternative splicing is really part of the study to understand the gene expression, or the whole, you have to understand the whole story so that you can see what you're going to do in the next step. And the next gen microarrays, in this example, Clarium D, actually enabled us to understand the mechanisms of drug resistance, and this will further enable us to use in the future targeted therapeutics, which drugs we should choose. And I gave you two examples from each of the preclinical research studies for the utility of this technology. And as a take-home message for the technical aspect, I would say the advantages of Clarium D for our RNA-seq is, first of all, it's a probe-based technology, because it's the coverage is extensive with multiple probes per target, and then it includes huge number of coding and non-coding. It is one assay to go compared to the depth coverage of the RNA sequencing. And the bioinformatics is relatively simple, especially a molecular biologist like me. I would like to go back and forth with my wet lab and then analysis at the same time. Whatever data I have, I can immediately go and analyze based on the new data, very user-friendly for the biologist. And you can, it covers all the transcriptome analysis, high, low abundance, non-coding, or alternative splicing, it doesn't matter. It's a multi-purpose technique. And another, as I mentioned previously, I like to see that it is you can go directly from the software to the other browsers, UCSC or Ensemble or others, so that you can understand what this region really, what this exon change or location really means in terms of biology of the disease. And of course, it's easier for the adaptation to preclinical studies like I showed you, and it will be a potential clinical use in the future. And the last but not least, it's very cost effective. With this, I would like to thank to my collaborators and lab members, and of course to Thermo Fisher Scientific and our funding sources. Thank you.
Thank you so much, Dr. Gökmen-Polar, for that informative presentation and your important research. We will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask audience members, please do so now. Just click on that green Q&A button at the lower left of your presentation window, type that question into the box that appears on the screen, and click the send button. So let's take a look at our audience member questions that we have already coming in. Our first question, Dr. Gökmen-Polar, is what are the similarities and differences between coding and non-coding RNAs to study in cancer research?
Great question. Of course, there are similarities and dissimilarities. So in terms of these similarities, they are mostly transcribed via RNA polymerase II. They have polyadenylation, and they can undergo alternative splicing because they have exons. On the other hand, there are challenges with the non-coding RNAs, especially because they mostly exert a regulatory role rather than enzymatic functions. So that makes it no enzymatic assays available as readouts, or because you don't have a protein expression, you can't go by the antibodies and look at the expression, and there are hard to find readout assays. Another challenge is they are expressed in low levels. So for knocking and knockdown, usually most of the promoters are very much designed for messenger RNAs that where high expression is mostly desirable. And therefore, to exactly mimic the same conditions for the long-coding RNAs, non-coding RNAs are a little bit more difficult than the messenger RNAs. However, one way to understand their role will be to determine their cis and trans mechanisms. So you can use computational assays and look at the exons that is affected and the motifs, what kind of RNA motifs you find there. These are actually lead to you further understand what kind of pathway you can focus, and you can also look at the trans mechanisms, what other distant pathways are affecting those RNAs. These are the ways to study.
Thank you, Dr. Gökmen-Polar. Our next question from our audience is, what would you suggest as a next step following identification of a splice variant?
Well, it all depends where the splicing occurs. So it may affect the cassette exon or 5' 3' region or any other region, but the most important thing, you have to connect this region whether it's an important region in terms of biologically. So therefore, which I give the example for the with the software that once you know where the location is, and you can connect this to other data sources like Ensemble or UCSC, and you can understand the change further. You can connect to the protein domains. Let's say one exon included or excluded. If it affects a particular protein domain, that means that it alters the function of the gene. And as I give in the CD44 example, so including the exons affect the protein structure and domain, and therefore at the end it also alters the function. Another thing, that mutations can appear, and mutation can cause splicing, and also that affects further the downstream biological effects. So it all depends where it happens.
Thank you, Dr. Gökmen-Polar. And I want to thank our wonderful audience today. We have time for one more question. Our next question, Dr. Gökmen-Polar, is what are the methods to understand the role of alternative splicing?
That's a great question, but a difficult one. Of course, you can do the same thing that we do for the messenger RNAs. You can knock in or knock down the particular exon or exons. But this is, of course, more challenging because you need to develop mini genes, meaning that a mini gene is a minimal gene fragment that includes an exon or the control regions necessary for the gene to express itself. So you can develop the same way as the wild type or the major form of this, and you can also develop more complex mini genes, including multiple exons. That's one way. Other than you can introduce the whole transcript that is missing or excluding that exon only or exons only. So these are the multiple ways that we can analyze specific exon related assays.
Dr. Gökmen-Polar, thank you so much again for your presentation today. And I also want to thank LabRoots and Thermo Fisher Scientific for making today's educational webcast possible. Before we go, I want to remind everyone that today's webcast will be available for on-demand viewing through April 2018. You'll receive an email from LabRoots letting you know when the webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thanks for joining us. We hope to see you again soon. Goodbye.